biologist as well as a microbiologist, and so she was a co-supervisor of this project. And if you look at Malaysia, okay, Malaysia actually occupies two land masses. This is the peninsula, which is attached to the mainland, and a part of Borneo Island also is under the Malaysian government. And so we were able to get samples from both these areas and investigate the subspecies question. As you know, getting samples, genetic samples from other countries and working on them in your lab can be very, very difficult. And so we had to work with what we had. And because Borneo is a subspecies, we have a subspecies over in Borneo, and then here this is considered the mainland subspecies. We wanted to look at the molecular evidence, okay, for the subspecies separation. So the first thing that Shireen did was she assembled the mitochondrial genome of a Malayan sun bear, and she developed mitochondrial markers, genetic markers, that she was going to use to look at evolutionary history. Okay, and then she also developed nuclear microsatellite markers so that in the future, if somebody wanted to look at population size and get at questions of where you needed to know individual identity, known as mtDNA, really useful, okay, to look at questions related to evolutionary history. Where did these bears come from? How long ago was their separation? Okay, so for example, the Sri Lankan sloth bear and the bears on the Indian mainland, they're considered two different subspecies. With mitochondrial DNA, you can actually look and see, is there a separation between the two? How long ago was that separation? Okay, so we were looking at this kind of question. And by the way, this has not been looked at for sloth bears. Okay, so think about it. Okay, the other thing is that you can look at population genetic questions, okay, such as interspecific diversity and intraspecific diversity, looking at the subspecies separation, okay, that would be intraspecific diversity. Gene flow, okay, and geographic origin. So for example, if at the customs they confiscate some body parts of a sun bear, you can use mitochondrial DNA, and you can also use nuclear DNA to actually determine the origins. Which part of the world did that bear come from, or which part of Southeast Asia did that sun bear come from? Okay, so lots of forensic applications as well. Microsatellites, okay, these are extremely variable. These are nuclear markers, and very useful for looking at individual identity and kinship. Okay, so I'm mostly going to talk about mitochondrial DNA. So Shireen, she mapped the genome of a peninsula sun bear. You need a fairly good genetic sample for this, okay? You can't use degraded DNA because you need fairly intact DNA to get the full mitochondrial genome. And she then downloaded mtDNA genomes from the internet. So these are in GenBank, okay? There's a full database of any mitochondrial genomes that have been published on bears will be uploaded into GenBank. And she basically compared the Peninsula Malaysian sun bear with what was available in GenBank. And in terms of what is available for sun bears, it's quite low, okay? Quite a little. And this is where, if you look at the phylogeny, okay, so we have sloth bears over here, polar bears, brown bears, and here are a group of sun bears, okay, in this area. And the Malayan sun bear that she sequenced, the mtDNA genome, is highlighted with a red box around it. And I'm going to zoom in on that. Okay, a couple of other things that I wanted to mention here, okay, in the phylogeny. The giant panda, whoops. Okay, the giant panda, okay, it's considered a bear, okay, and it is considered a very primitive kind of bear, and so you see that it branches off very early from the bear evolutionary tree, and then the next very old branch is the spectacle bear, which is right here. Okay, just wanted to point out that. And then all these other bear species, okay, they're known as the ursine bears or in the subfamily Ursinae. And they are fairly modern bears. And the sloth bears belong to that group. So looking at sun bears, okay, at what is available, we noticed that we had, this is the peninsular bear, which is the same subspecies as bears from the mainland, and the only bear that we knew the geographic origin for was a sun bear from Yunnan, China. And it looked as if there was a separation here, quite an old separation between the peninsular bear and the Yunnan bear. We don't have any Bornean bears, mitochondrial genomes in GenBank as yet. And so this was somewhat surprising because we would have expected the peninsular bear to be quite close to the Chinese bear, right? The Chinese sun bear, because they're the same subspecies. So this gave us a little bit of cause for wondering what's going on. And then we started looking at the literature. And this is a paper that was published by Kumar et al. In 2017, I believe, and they pointed out a similar separation, but they thought that it was between the Bornean bear and between the mainland bears. Okay, they said there was a historical split and an old split, and these two bears had very different evolutionary trajectories. But they didn't really have a Bornean genome to look at, and so they imagined that the split that they were seeing was between the mainland bears and the Bornean bears. So let's have a look at the region where peninsular bears come from. Okay, so this is the Malay Peninsula from where that genome came from, and this entire area is known as Sundaland. Okay, or the Sunda, it's a unique biogeographical region. Includes Borneo, Java, Bali, Sumatra, Indonesia. The climate is tropical, and the thing about Sundaland is currently, okay, all these land masses are separated by ocean, but if you look at evolutionary history, okay, during the Pleistocene and so on, there were many periods when you had cold spells, okay, and you had ice ages, glacial maxima, where there was a lot of ice that was formed on the Earth, and the sea levels dropped during that time, exposing the Sunda Shelf. So it's basically a tectonic plate where the sea is quite shallow, and so most of the time, the Sunda Shelf would have been exposed, okay, allowing for gene flow or movement amongst all these regions that are on the shelf. So animals should have been able to move around here. So a couple of other things. This paper, we looked at this paper as well, and this is the Malay Peninsula over here. And these scientists looked at mammals' diversity, okay, in these different blocks. So here we have the Indo-Chinese mainland, and then we have this area, this very narrow area between Thailand and the peninsula, which is kind of like a narrow neck. And what they found was that in this region, okay, where you have this very narrow area, there were about 30% fewer species, okay? That, if you look at it geographically, is between 6 degrees and 13 degrees north. And even species that were very widely distributed, okay, across this entire area, there were big gaps in these two blocks, okay? And this is where those two areas correspond to this, and it's called the Isthmus of Kra, okay? which is a very narrow neck between the peninsula and Thailand. And they attributed this to sea levels that very often rose and extirpated a lot of the life that was on that region in this very narrow area. And this basically gave rise to this somewhat depauperate fauna on the area. And so basically what I'm trying to sort of lay the groundwork for is there's a possibility that Sundaland as a whole was separated from the mainland, okay? And that we might be looking at a different sun bear subspecies that is found within Sundaland, not just within Borneo. So if we look at glacial cycles, okay, over the past 450,000 years, okay, a lot of the time, okay, the Sunda shelf would have been exposed and allowing for gene flow within the Sunda shelf. You had very different climatic conditions here, okay, aseasonal, warm, humid conditions over most of the year. This is warm, tropical climates. Okay, now we look at the distribution range of the sun bear, which is highlighted in yellow here. Okay, the Malayanus, HM Malayanus, okay, is supposed to occur in these three regions, and then we have the Bornean subspecies over here. Okay, what I've highlighted is the Sundari part of sun bear range. Okay, so what we did was Shireen designed 3 sets of mitochondrial markers. What happened? Here we go. Okay, so she used the whole mitochondrial genome to identify the D-loop region. Okay, and including adjacent areas, okay, of the D-loop region. And she developed a set of overlapping markers, 3 pairs of overlapping primers, so that it would cover a fairly sizable part of the mitochondrial genome. Okay? Now, the reason that she designed 3 overlapping sets of primers is because when you amplify a smaller piece of DNA, you can actually use DNA that is coming from somewhat degraded sources. You don't need a good muscle sample or a blood sample to find mitochondrial DNA. And so these were the primers, and each one of them was about between 500 to 760 base pairs. Okay, so much easier to amplify DNA amplicons of that size, okay, fragments of that size, than a big fragment. Okay, because what happens when DNA leaves an animal, either on the scats or in the form of blood, it starts degrading and breaking down and fragmenting. Okay, and so if you are working with fragmented sources of DNA or degraded sources of DNA, having a smaller a primer that amplifies a smaller fragment is to your advantage. Okay. So we ended up with 82 genetic samples. Okay. And we had 28 from the peninsula, and we got 36 samples from Borneo, from Sabah, from different parts of Sabah. And then we were able to get 18 samples from Asiatic black bears and sun bears, and he was able to go to a sun bear captive facility and get hair samples. He basically, what he did was he offered the bears some honey at the edge of the cage, and when they came to get the honey, he just reached in and grabbed some hair and pulled it. And he got some hair samples that way. And Shireen went to Thailand, to his lab, and she did the DNA extraction. They sent the amplicons for sequencing in Thailand, and then we were able to get the data, and that's how we were able to manage this. Okay, so we basically then analyzed the sequenced data, and let's have a look at this. Okay, we looked at the phylogenetic tree, and we measured some different aspects. We looked at haplotype diversity within each population, and we considered Peninsular Malaysia to be a population, the Sabah bears to be a population and the Thai bears to be a population. Okay, diversity within each population, and then we looked at FST, which is a measure of how different these populations were from each other. Okay, so if you get a large FST value, what that suggests is that the two populations have diverged significantly, meaning that they have been separated from each other for a fair amount of time, okay, and they've gone on their own evolutionary pathways. So interpopulation variation, if you're, and within population variation, I put this picture in here, okay, so if you're looking at variation within a population, and these are bighorn sheep that are on a mountain side or on the side of a mountain. These are animals that live on mountains, and they very rarely move between mountains, so each mountain can be considered a population, okay? So if you look at genetic variation within that, you would be measuring haplotype diversity if you're looking at the mitochondrial DNA, okay? If you're looking at how different are the goats on this island relative to this island, then you would be measuring FST, okay, which is between population variation, okay, and sometimes you have complements of alleles that are unique to certain populations, and that's going to make FST higher, okay, and make those populations more unique. Okay, so I know that the text is going to be very difficult for you to look at here, but in red, okay, the red branches correspond to the Bornean populations, and the green branches, which are over here, correspond to the peninsular bears, okay? And what we found that was really interesting here is that about 1.5 million years ago, there was a very deep split between the mainland and the Sundaic populations. So Sundaland bears separated quite early from the mainland bears based on the mitochondrial DNA, okay? And then following that, there was a more recent split between Bornean and peninsular bears, okay? And so I'm just pointing out the main things over here. None of the Bornean and peninsular bears are in this little branch here. These are coming from Thai bears, okay, that were from the mainland. And what was interesting is that there were some Thai bears that fell into the peninsular clades and into the Bornean clades. And so it looked as if bears from Borneo and the peninsula had historically moved upward from Sundaland back into the mainland. And that's the nice thing about mitochondrial DNA, can help you to sort of identify some of these patterns. So if we look at the diversity, haplotype was probably originally, the sun bears would have been a large outcrossing population, okay? And so the N corresponds to the number of bears, okay, from each of these populations, Borneo, Peninsula, Malaysia, and Thailand. And this is the haplotype diversity, okay, the different number of different haplotypes. And if we look at FST, how different they were, in fact, significantly different, okay? If you compare Sabah and the Peninsula, the FST was about 0.5, which is quite high, and significant differences between Sabah and the Peninsula, Sabah and Thailand, and between the Peninsula and Thailand. So these are well-differentiated populations. Okay? The interesting thing is that Thailand seems to be a place where you have a fair bit of genetic admixture. And so there are 3 mitochondrial lineages, well-differentiated mitochondrial lineages within Thailand. So what do you think was going on here? Okay, so we saw this very ancient split, okay, where we had bears from the mainland splitting from the Sundaland bears. And so what we think actually happened is that a population, okay, 1.5 million years ago or so during the mid-Pleistocene, during glacial maxima when the Sunda shelf would have been exposed. All these light blue areas, which are shallow seas, would have been land during that time. That we had the spread of bears from this area down into Sundaland. Okay? And then what happens is when you have the melting of the glaciers and the sea levels rise, these populations in Sundaland are separated from each other and they evolve in isolation. And so they become more different from each other. Okay? And that would have accounted for the differences between the peninsula and the Bornean bears. Okay, but a more recent split. Now, what accounts for the Thai bears appearing in the Bornean and the Peninsular clades? And what we think must have happened is that during these times when the Sunda shelf was exposed, that you had some back migration from the peninsula and from Borneo. Had no mixing between Bornean bears and Peninsular bears. Well, there was a paper that was published in 2020 where they looked at bat feces and bird feces found within caves, fossilized feces, and they did a radioisotopic analysis to look at what was contained in these feces. And from that, they were able to decipher what the habitat was like, okay, during these times when the Sunda shelf was exposed. And so what they found was these areas that are highlighted in orange, these are coming, okay, these areas were savanna. Most of what was found in the scats, in the bird scats, were the remnants of grasses. And so and there's some additional evidence coming from this paper as well that much of this area in Sundaland, okay, that was between Borneo and the peninsula and Sumatra would have been a savanna belt, which means that animals that are very dependent on forests would not have been able to cross those savannas. And so this would have kept the peninsula bears and the Bornean bears separate. And so there were barriers to gene flow, okay, which is mixing of these two populations during times when the Sunda shelf was exposed. So that explains, okay, to some degree, why you don't see a mixing between Bornean and Peninsula bears. Okay, and so the question still remains that Borneo is somewhat unique can be up to 3 months, okay? That a leech that has fed on a sun bear 3 months ago will still have traces of sun bear DNA in the gut. Okay, so this is an ongoing area of research. These are potential areas of research as well. For you, you don't have to do it on bears. You can try it. Leeches are not found in habitat that sloth bears occur in, but there are other species that they feed on that you might be interested in. Okay, and iDNA is a way at monitoring some of these populations, and you can get DNA of such good quality from a leech that you can also do microsatellite work. Okay, and then dung beetles is another thing. So dung beetles that feed on sun bear dung, we're looking at this as well, doing some control studies to see for how long between feeding on sun bear dung and DNA extraction, for how long can you get sun bear DNA from a dung beetle? And it's usually maximum of 3 days, okay? Usually 1 day later has degraded. Okay, so something that's pretty critical, okay? Asia is developing rapidly. We are in some of the tropical Asia in particular is in one of the most densely populated parts of the world. And so wild species, okay, wilderness is suffering as a result of this, and animals like bears and large carnivores are being rapidly depleted. And so monitoring these populations, developing efficient and feasible ways to monitor these populations is really, really important. And something that I want to emphasize here is that a one-time density estimate is not useful, okay? It is the changes in density over time that is what we need to look at, okay? So if populations are staying stable, we don't need to worry about them. If they're increasing, like they are in North America as well as in Western Europe, bear populations there are expanding, okay? Then we don't need to worry. But if they're declining, then we need to worry. And we can only find out if they're declining by monitoring them, okay? In many parts of sun bear range, you have good habitat, but you have no animals because the poachers have removed them. Vietnam has empty forests, okay? You have rainforests, you have no animals. And so finding ways to monitor these animals is really, really useful. Now, these are a couple of monitoring methods, okay, where you can look at changes in geographic range. This is in order of decreasing geographic scale, okay? You can map the distribution just by, you can use survey methods. Interviewing hunters, for example, are bears present here? Have you seen bears in the last year or seen bears sign in the last year? Bears, because they're large and they leave very conspicuous footprints and scats that are readily identified. Scats are the feces, okay? If you have worked to recognize a bear scat very easily, a hunter knows how to recognize bear footprints and bear feces. If they have seen it, that's an indication that in the last 2 years or so, you have had bears in this area. You can map a distribution this way, okay? And if you're covering large areas, that's one way to do it. You can look at changes in occupancy. That usually takes a bit more work, okay? Because you have repeated visits or repeated site visits, or you have to have a lot of replicates, okay, within a site so that you can come up with a probability that the site is occupied. So that's another way that you can do it. And then finally, at the smaller scale, is looking at changes in density. Okay, and this is the only method that will actually give you numbers. Okay, all three of these methods are good ways, okay, to monitor populations. Changes in density will tell you how many bears are there in this area, okay, or what the density is. And for that, you need to be able to identify individuals, which is quite challenging with bears because unique identification of bears is not easy. So if you can get a good picture of the chest mark, which tends to be quite variable amongst individuals, that's a way to do it. Or if you can get a genetic sample, okay, like from a scat. And genetic identification from sloth bear scats has been done, and there are primers, microsatellite primers available to identify individuals. Okay, India has done this work. You also need a very rigorous study design, okay, to get good estimates. And then you have an open population spatial capture recapture models, which are right now the gold standard for estimating densities. And so I'm going to stop here, and thank you all very much. This is my group of my colleagues and students in Borneo, in the rainforest at Tabing Wildlife Reserve. I thought you might like to see the size of some of those big rainforest trees, take you back to Singaraja. And so if anyone has any questions, I'm open to that right now. Thank you. I hope I didn't run over time. We have, okay, all right. How much time do we have?
1 hour.
Okay, all right. Any questions?
Thank you, dear ma'am, for this very wonderful, informative and knowledge-filled talk. It was really inspiring, and since I know about the curriculum, I only know about the curriculum of our degree program, which is Environmental Science and Natural Resource Management, that we do offer from the Department of Natural Resources, and I think there are students coming from various other departments as well. So, on behalf of the department, I'm really sure that our students gained a lot of information and education from your talk. So please, dear students, this is the time for you to have your questions. So this opportunity will never come soon again. So please make sure that you questions and queries. Please.
I hope some of the technical details on the genetics and stuff wasn't, I didn't realize that we had some non-biology students in here as well, but there's no question that is unsuitable, okay? So if you have a question about bears or sun bears or sloth bears, please go ahead and ask.
I'll take the first question. I have to pardon me, and there's an emergency and I have to run out. But just this question has nothing to do with sun bears.
Yeah.
Because since your work, there has been no substantial work on the Sri Lankan bear.
Right.
So what is your recommendation? There are a lot of people who are interested in doing field work. What are your suggestions? So what do you think that needs to be done about Sri Lankan bear in terms of research? Because we are thinking of starting something. So I would like to hear from you first. What needs to be done? What are the research priorities we have right now?
So I'm thinking, the way when I started with sun bear research, in Malaysia, I was new to the area, and I had a very busy job as a faculty member. But I had students who needed research projects, and I often started with undergraduate research projects, honors thesis projects, and developing fairly simple projects. And one of them could be from your, if you are from a part of the country that has bears, maybe interviewing the local people to find out about attacks by sloth bears. Sloth bears attack a lot of people, and interviewing, developing a questionnaire to interview them about the circumstances of the attack. You can also look at presence or absence of bears. You can create a grid, overlay that over a part of an area that you think you can cover, and map bear presence and absence in that area based on interviews or based on sign, on bear sign. If you have access to a lab where you can do some PCR work, some simple projects might be to just take some samples from sun bear feces, bring them back to the lab, and see whether you can extract the DNA and amplify it with some of the published primers. Some of this very baseline work is publishable, okay, because it hasn't been done before. So don't dismiss these as, you don't have to have a big groundbreaking project, okay, at the undergraduate or the master's level. You can pick something simple, and if it's new information, it's good information, okay, we need that. And it can be simple, fairly simple. What else can you do? If you have access to remote cameras, okay, you can perhaps do a habitat-related study with sloth bears, or you can do what Thailand did. See whether you can develop a way for the sloth bear to stand up in front of the camera and show its chest mark. And if you can get enough pictures like that, okay, that is, and get a measure of how many times you were able to get good photographs. These are all forms of data that is useful for developing a good sound method for monitoring density of sloth bears. Okay? Hair samples, barbed wire traps. Okay? See, the sloth bears have long fur, and it's very, how to say, it's coarse fur. So I can see that it will snag in barbed wire quite well. How are you going to attract them into an enclosure that has barbed wire around it? Try some different things. When I was capturing sloth bears, I had to try a lot of different things, and I found that bees honey worked quite well. Okay? And then you have to think about, how do I put the bees honey there without the ants getting to it? Okay? We used to use a little plastic jar with a wide opening at the top, and then we covered that with cloth, and we tied the cloth tight around the neck, and that kept the ants from getting inside and eating the honey. And then we poured a little honey on top of the cloth as well, so that the bear would get the smell of the honey. And get good wild honey, okay, from the jungles. Don't go and get this commercially imported honey from Australia or wherever. Get the wild honey. We used to go into Hectipola and sometimes into Pallegama to get honey for the traps.
We have beekeepers in this region. Yeah. So it's easy to find good honey in this area.
Can find good honey. Yeah. And you know, the wild honey just has a nicer smell to it, I think. Yeah.
Okay, I open the grounds. Questions? Problems?
Madam, I have one question. I think it's a very interesting talk. Do they have their own territory?
They have a home range. So there's been only one radio tracking study on sun bears, and it was done by Sute Wong in the early 2000s. And they have a home range, but just like with sloth bears, the home ranges of males will overlap with that of other males and with that of females as well. Female home ranges overlap, and they overlap between sexes and within sexes. Yeah. Most bears tend to be, you know, they have an area that they will move around. So the home range, if you define it, it is the area that an animal generally uses in its day-to-day life, right? There might be a few forays where it goes out very far away to investigate something, and generally that isn't considered a part of the home range. And you can statistically actually get at the boundaries of home range. You have methods like the adaptive kernel and harmonic mean to measure home range size. But you have to be able to track an animal if you want to measure the home range, right? And so you have to capture and put a collar on it and release it. And then you have to get the data. It's very labor intensive, and it's not for the faint of heart. You will have no life when you take on a wildlife project.
You can't get out.
No life other than your research. But that's true for, if you're a biology major, you have no life anyway, right? Yeah. You get out of bed and you roll out of bed and you come to lecture in your pajamas because there's no time to change. Okay.
Phylogenetically, what is the other group of animals that closely related to the bears?
The mustelids, I would say. So we have the canids. Very often we use the canids, a wolf, grey wolf, as an outgroup. But the mustelids are quite close. Phylogenetically, the the closest carnivores that are related to bears are the seals, and it's one group of seals. They're aquatic, but they're carnivores. Okay, but they are the closest relatives of the bears.
Is the leopard seal?
I think that's a eared seal. It's either the eared or the earless seals that are closely related to the bears. And I think it's the eared seals, seals with the ears, external ears. Might be wrong though on that. Yeah. It's one of the two.
I have a technical question.
Yeah.
Did you sequence those microsatellite DNA?
Yes. I think we use NGS for sequencing the
How do you
these.
manage with that non-specific amplification?
Oh, the microsatellites. We didn't sequence. We just ran them on agarose, and we knew that. So we cut out some of the bands and we sent it and we did Sanger sequencing to make sure that it was the target area, and it was. So we used Sanger sequencing for the microsatellites, but because we were not really doing any major study with that, for NGS you need a lot of samples, right, to make it financially valid. So this is just testing. So we use Sanger sequencing, and we made sure that we got the target sequence by getting the base sequence. Yeah.
So you did a gel purification with the exact.
Yes, yes. The Malaysian students at Sunway are very good with the genetics. Yeah, because the program was biotechnology, medical biotechnology and biomedicine, so they do a lot of lab work. But when I offered projects, I had lots of students wanting to do the wildlife projects. Yeah.
Maybe two questions. Why are they called sun bears in the first place?
Somebody asked me this question. Okay, so why is a sun bear called a sun bear? And you'll get different answers to this, okay, depending on who you ask. The common, I would say, belief is that because the blaze, the chest blaze of the sun bear has a yellowish color to it, that it's called the sun bear. But the very old literature that talks about the classification of the sun bear talks about it as the bear from the hot, warm parts of the world, hence the sun bear. Okay, and I think that is the correct reason why it is called the sun bear, why it came to be called the sun bear. Yeah.
Then why aren't our bears called the sun bears?
So unfair.
Yeah, so unfair, right? This is the hottest part. This is hotter than the
So when the sloth bear got sent to the first specimen, okay, that got sent to the British Museum, they didn't know where it was from. They thought it was a sloth, a true sloth. These are the South American animals that hang upside down from trees, and they have very long fur and they have long claws, and they have no teeth, they have no incisors. And because the sloth bear is missing some of the incisors, and it has this long shaggy coat and these long claws on the feet, they classified it as a sloth that looked like a bear, so they call it Bradypus ursinus, or a bear-like sloth. It was later corrected. It's a true bear, okay? And for a while they thought the bear came from Africa, but it's not from Africa. You know, these things come in ships and they get thrown in with all other specimens. They had no idea where the bear was from, so they call it Melursus libius for a while, which is from Libya. That was the bear. And so it's gone through several names, but the common name now is sloth bear because it's a bear that looks kind of sloth-like. Okay?
But when we heard that word sloth, it's comes to mind like a very lazy animal, right?
Yeah, this is true, but it's not, right? Sloth bears, if a sloth bear is coming for you, you cannot outrun a sloth bear. People who've been attacked by sloth bears said that when the bear charged, they said all they could see was dust and flying leaves. They didn't even see the bear. Until they had contact with it very fast. They can be very fast.
Did you find any records related to their fossilized
Fossil history. You know, I had forgotten and I don't have the paper anymore. But I think Viraniagala had an old paper, a short note about some bear fossil, fossil bear. And it may have been coming from a cave in the wet zone. But he didn't think it was a sloth bear. He thought it was a sun bear. And I've tried looking for the paper and I don't have it any longer. So there is some information from the old archaeological papers, I think.
Any questions from the students? Let me make a very short announcement because Professor Shamila and we have some plans to start some research in the near future. So if anybody is interested, come and talk to us, the department, and see what we can do. That's the first thing. And she also has plans to come to Sabaragamuwa, which we are really privileged on yearly basis, like annual basis.
Just as a visiting
professor to come and teach about it. So we are trying to organize it from next year onwards, hopefully, if things get sorted out. You have that opportunity. It's like something really great for us. And also the statistics boot camp, right? Would you like to tell a little bit about that?
So we are planning to hold a boot camp next year at Sabaragamuwa, and this is something that I have worked with. It was initially run by Mike Meredith and his partner Numbang Juat, who are based in Kuching, Sarawak, and they have a small NGO called the Biodiversity Conservation Society of Sarawak, and they run these boot camps, and they're capacity building boot camps to train young scientists who are working in wildlife conservation how to analyze their data, okay, using some of the most current methods. And the nice thing about the boot camp is it's very hands-on. So it's not somebody here standing and talking about stuff. You will come with your laptop and you will learn how to analyze the data. It's hands-on work and it's quite intensive. It takes 10 days and it's not for the faint of heart. Spatial capture, recapture models. And I might need to use some kind of, what do you call this, habitat suitability modeling, okay? Species distribution modeling, something like that. This kind of boot camp would be useful for you. And we use open access software, Program R, which is something that is free. You can download it into your laptop. And I don't know that we will do any QGIS, okay? But it will be mostly stats and with Program R. And you also get a chance to talk about your project that you have planned and get some feedback from the other participants. So it's really suitable for a small group and suitable for individuals who are going to need to analyze wildlife data. Okay, so if you work in a lab, you're doing something that will be useful for you, then sign up for it. Okay, I think there's a great need amongst the tropics in our developing countries, India, Sri Lanka, Malaysia, Indonesia, for students to get this hands-on experience with analyzing data. Okay, and so this bootcamp offers that kind of opportunity. So if you think this might be something that you want, sign up for it. So, Prof. Inoka, this is, I think your guidance in this might be also very useful because, you know, for a student like Terence who was doing the leach work, and it was very experimental, very controlled experimental work. He doesn't need to know about occupancy models and SCCR. So a boot camp like this would not be suitable for him. But for Thailand, Y Park, Wilvia, many of my students are working with cameras, remote cameras, and or any kind of capture, density estimation, and so on. These types of boot camps would be useful. Okay, yeah.
Okay, one more question.
Okay.
Don't take it as a complaint, but
Yeah.
I think in the last week you had a speech at the MICH.
Yeah, yeah.
Right, on slot bears. It's on the screen. But today you talked about sun bears. Why?
I wasn't sure whether the same people would be at the WNPS lecture. And I was in the United States at the time, and the slot bear work that I did in Sri Lanka was done a while ago. Okay? What we're doing on the sun bears is more recent. And so I think it's important for you to look at what's being done more recently so that you can apply it to questions that you might want to ask. Okay? The species doesn't matter. It's the methodologies. Can I take the methodology? Can I apply it for a question that I want to ask? Whether it is bears or leopards or mongoose or barking deer or some kind of bird, okay? For my PhD, I worked on raccoons, which is a small carnivore. My major professor, who ran a 30-year bear project in the Great Smoky Mountains, Mike Pelton, he worked on mice for his PhD. Okay? So switching organisms happens all the time and species, but the methods and the theory and all that can be applied in lots of different ways. Okay? Answers of, would you have preferred to have heard about sloth bears?
Yeah, I mean, since it's there in Sri Lanka, right? And I thought that students might be more interested because they can start learning on something. Yeah, yeah, sure.
I promise to do a sloth bear lecture next year, next time, okay? And I think you will find it very interesting. There won't be any genetics, okay? Yeah. Today they got a lot of genetics.
Species, what she said about species, I'm just speaking freely because these are most of them are my students. I worked on rats for my undergraduate research, toxicology. My master's was on elephants. My PhD was on dung beetles. Now I'm working on leopards. This other species. Right? As long as you want to be in the field, you enjoy what you're doing. I didn't enjoy working on rats though, but I used to.
Yeah, I also do work on invasive snails and otters. So kind of spread a bit thin. Yeah. All right, everybody. Thank you very much.
No questions anymore?
I'm sorry about the videos.
Yeah, there's one more question.
That's not
Oh, we have a question. Yeah.
Madam, maybe these areas like systems would be added since they are living in an near earthquake belt.
Say that again.
Wouldn't their existence be affected by the earthquake since they are living in near an earthquake belt?
This is true, right? Because that area near Indonesia is there's two plates that are coming next to each other. I think it's quite localized, where you have the earthquakes, and then sometimes you have a tsunami, right? As well, tsunamis as well. The bears are deep in the forest. I remember that one area that has sun bears was affected by the earthquake, and it's possible that there might have been a few individual sun bears that might have been lost. But I think overall, earthquakes don't seem to have a major effect on the populations, at least not one that is measurable. Okay? Yeah. Thank you.
Ma'am, have there been any citizen science sort of relatedness in your work in Malaysia, Sarawak, and those areas, indigenous communities helping in conservation efforts?
With some of our work, yes. So my student that works on the otters, he works for the Malaysian Nature Society, which is like WNPS, is a very old conservation organization. So they work with local people a lot, and so his field assistants are coming from local people. And then one of my students, Wilvia, is from the Iban headhunting tribe, and she did her master's with me. And a lot of our field assistants come from the local tribes. Yeah, they're very good in the forest. Yeah. There is some citizen science work done in the peninsula with by the Malaysian Nature Society, especially with fireflies. Yeah.
One small comment. When I was an undergraduate, craving to do wildlife conservation, there were very few women in the conservation field. Very few women who had to look up to, and for me, she was one of them. Like Professor Savitri Gunadilekha was working in the forest, and she was working on bears, and sort of gave us motivation for all the girls out there. Some of you are saying that is it okay for a girl to go to the conservation center? Is it hard? Just look at her. And that's all the motivation you need. Yeah.
Yes, when I worked on sloth bears, I was one woman with a team of 3 other men, and it worked out very well. It worked out just fine. Yeah. And so, you should be able to convince your parents, I think, in this day and age, that it's quite safe for a young woman to be out in the field without a chaperone. Thank you.
So with that, even though we did not have questions from you all, I'm sure that you gained a lot of information and a lot of knowledge from her talk today. And you must be having numerous things in your mind right now. And please don't hesitate to communicate with Madam, maybe through emails now. Through emails, or you can talk to Inoka Madam or the department or one of us, maybe. So let me thank you formally on behalf of the 9th International Conference of Sabragam University of Sri Lanka and the Department of Natural Resources, Faculty of Applied Sciences. And we were lucky to have the presence of both sirs, Dr. Ranga Vimalasri, the chair of the conference, and Professor Deni Charala, head of the Department of Natural Resources today here. So thank you again, ma'am. It had been a very nice experience, and we wish to see you in the near future as a visiting scientist in the Sabragam University of Sri Lanka.
Thank you all for coming. It's been a pleasure for me to come and talk to my fellow countrymen.
And thank you, team, for putting this together. Samira and Chandi and everybody on the conference. Thank you so much. Because I planned this to have it at the faculty, but you took it to the next level. Thank you so much.